Hi everyone, thank you for joining us for the final Back to the Basics webinar. Today we're going to talk about some protein tools, and you'll get to hear from Roshni, who will introduce herself in a couple of slides. This is the final section of the Back to Basics series, and all of the ones that have already happened will be put up on our BV-BRC YouTube and in the workspace for this webinar series. If you don't already, please follow us on LinkedIn. So just a quick introduction. My name is Annika Priya. I work on the BV-BRC project on the viral team. I am the outreach coordinator. So if you ever have any questions, like a workshop, feel like the resource isn't working how you expect it to, please reach out to me and I'll try to problem solve or send you on to the right person to help you, which could be someone like Roshni or someone else on our team. So I'll let Roshni introduce herself.
Hi, I'm Roshni. I work as a bioinformatics analyst for BV-BRC. So a little background about myself. I have a master's in biotechnology from India, like St. Xavier's College. And then like in US, I did another master's in bioinformatics from the San Diego State because I felt like I didn't know anything about programming. So that was the next step. But I'm looking forward to sharing some tools with you from the BV-BRC.
Awesome. I'm really glad that I got to have Roshni join us today because I feel a lot less confident about the tools that she's going to talk to you about today. So I think we'll all learn a lot. If you've been to one of these webinars before, this slide is probably familiar, but I just want to orient everybody with the BV-BRC. So we have a mission to provide a one-stop shop for data about human pathogen and related bacteria and viruses, which exists in a scalable resource that also supports bioinformatics, large-scale data mining, systems biology, machine learning, and really our goal is to support the evolving needs of the infectious disease research community and serve as a paradigm for research data resources. So what that looks like in a little bit more detail is that we have a unified data model and real-time data integration processes. So we upload data from GenBank and other sources that we annotate and curate the metadata for to make it really accessible and searchable and usable for you. We also use a bunch of different tools, which if you're interested in kind of a broad overview of the BV-BRC in a lot more detail, I highly recommend the first one of this, Back to Basics, or looking at our help documentation. But we also do have a private workspace for you to do your analysis and share and collaborate with data, and then have API and command line interface and FTP for large-scale analysis and programmatic batch services. And then we have outreach and education, like us bringing you this workshop today. So I think I'm going to hand it over to Roshni to start talking to you about the services that we're going to cover.
So the services today, I will be talking about our Meta-CATS and the 3D structure viewer in BV-BRC. So I'll go into the phylogenetic tree also, but that has been covered by Anna in one of the previous webinars. So it will be just a touchdown, a brief to serve the purpose. But the services we are going to talk in detail are Meta-CATS, which is a chi-square test of independence on 2 sets, and the 3D viewer. So the objective today is I will take on 2 case studies and we'll take each case study and learn how to do analysis in the BV-BRC. Those are very small ones, but the first case study we'll work on is we'll explore a possible zoonotic or reverse zoonotic transmission event in influenza. And we'll try to decide whether the transmission took place from pig to humans, that is the zoonotic ones, or the other way from human to pig. We'll use Meta-CATS in this analysis. The next case study we'll do is a structural comparison case study where we'll compare the spike protein of the Wuhan strain with the XBB 1.5 strain. So let's look into details of that. The first case study, we talk about a possible zoonotic or reverse zoonotic transmission event in influenza. So a little bit background of what the case study is. So we are looking into the North Carolina state. The subtype is H3N2. and the collection year is 2022. So that's a lot of filtering of data. One type you can search data on BV-BRC is through the organisms tab. So you can go, like for my case, it's the influenza A. So you can click on the influenza A and go there and go through the genomes, and then you can go to the filters there and filter based on what you want. So you can filter based on the collection year, isolation country, state. You have additional fields here which you can include and do the filtering based on that. But today, because it's a lots of criteria, so I will filter, and I'm looking for the HA protein. So I'll filter the data based on the advanced search option. We'll go from the genomes first, because I want the genome also in case we need the genome for subsequent other analysis. So we'll start with the genome search. We have the influenza A virus. We need it from the state North Carolina. And then the collection year will be 2022. And we have a subtitle. Sorry, lots of details, but that's how you generally work on a case. And we have the segment 4 because the HA protein lies in the segment 4. So you can see I have filled up the form and then I do the search. And you can see we have 44 genomes. So the one way I proceed is I take all the genomes and save it to a group, which will be stored in my workspace. If you haven't signed up for the workspace in BV-BRC, please do it. It's very helpful for sharing. And also you can keep your data private if you want to. You can bring in your own sequences and also use all the tools available in BV-BRC and group them to save. But I won't save the genomes for now. What I will do is I will go to the proteins tab. So what I will do is once it loads, I will open the filters and search for only the HA protein because I am more interested in that. See, the protein option is not here in the filter. So what I'll do, I'll click on this and add the gene and the product. And you'll see this HA1 are fragmented hemagglutinin. But what I want is the full. So I'll select and I want the CDS and the HA. So I have my full leg HAs. We have 44. Remember, we had like 44 genome. So HA for all of those, and I select everything and save it as a group. I'll create a new group. It's a feature group because it's a proteins. And I'll say NC North Carolina HA 2022 test. So we have a new group now. So we add it. And now we have our data. So the first thing what we want to do is look at the phylogenetic relationship and see whether the transmission is from human to swine or swine to human. So next we'll go to the tools and services. We'll do a gene or protein tree. If you want to know about the phylogenetics tools in details, please refer to Anna's previous webinar. So we click on the gene protein tree and you can see there are lots of options here. So for me, it's the protein. So we click on the protein. I have a feature group already made. So my sequences are there. So I'll search for it and my feature group. And you see our NCHA 2022 test. I select that. Okay. And then place this plus button to add it to our list. And then we will keep it Raxamal, the LG model, and we'll select the output folder. Let's do it in feature group only. And the output will be NCHA 2022 test 3. You can see if you have a similar named file, it will give you a warning. And for this, I want to color the tree based on the host group because that's what we are trying to analyze, trying to see, trying to find out. So we'll go to the metadata options and see if our host is within the metadata table, which will be pulled onto the tree in the phyllo.xml file. But I don't see the host group here. So what I'll do is from here, I'll select the host group and then add it to the list with the help of this arrow. So you write your metadata field you want within the file XML file so that you can manipulate the tree. You select it here and you add it to this group, and you see within our metadata group, we have the host group and we submit it. So it's starting a job. You can go here in the job list, or you can click on the job list and it will take you to the list of job it's running. Hopefully it should be done fairly quickly. So you select the job from your job list and click the eye icon to view it. You can see a lots of files generated. The parameters are here, but what you're more interested in this view button, it will give you a consolidated result or your tree. So you click on this and sorry, sometimes this error comes. So the workaround is you just refresh your tree. It's a bug. We are working on it. So you can see the tree with the strains or genomes or HAs we have selected, but how do we know how it serves our purpose? So we will color this tree based on the host group. Remember, we selected is as part of the metadata to pull into the file XML file for this tree. So we color it based on the host group. And now you'll see. So the red are the human strains and the pink purple are the swine strains. And you can see there is a two clear demarcation, like one branch is for the humans and one branch is for the swine. But there are three sequences within this, which are swine sequences, but they lie within the human. So this swine sequences are more like human. So from here, the first hypothesis mostly it's the human strain which got mutated and is causing a disease in swine. So favor towards reverse zoonotic, that is transmission from human to swine. Okay, the next step you want to know that more statistical. This is just a phylogenetic analysis, right? So statistically speaking, we need more evidence. So for that, here comes Meta-CATS. So Meta-CATS, basically it does a chi-square test of independence on your groups. So like once you provide your groups, it will say statistically which positions are significantly differing between your groups. So currently it can do analysis up to 10 groups. So let's go through the parameters here. The parameters is the p-value. It's mostly standard. If you want to change it to something else, you can change it here. Again, the output folder you have to provide to store your results. So we'll give it a short name this time and say this. Okay, and then the input option, you have three options. One is auto grouping, where you provide a feature group which you have previously made from BV-BRC and provide that, and provide the metadata field here based on which you want to make a group. This is the most useful I have found myself. This feature group option, if you select this, you need to make your group separate. So you have to make two separate feature groups for label 1 and label 2 and provide that, or any other number of groups you want to make. And you have to provide it here and then it will make the analysis. The alignment file is you upload an alignment file, and the group file is you have to upload the metadata file for that alignment based on which you need to do that comparison. For my case, I want to do auto grouping. Host name, like based on the host field, that's what I want to compare. So host name is good. Otherwise, you can search. There are lots of options here from which you want to search. Then you have to select the feature group. So my feature group, I have the HA1 test and OK. And then you have to click this plus button to add it to our analysis. And now you can see the host group. You can preview the group as it is. So you have the metadata and the group it's providing. And these are our 44 sequences that we selected. If you want to delete something because there may be a one exception to your group, you can select it and delete that row. If you want to change the label of a group, you can do that. You can write the name of the group here. First, you have to select the entries, write the new name, and then click on change group. It should change the label for that group here. But for us, we wanted to do a comparison between human and swine. I can see those. it matches. So nothing to rectify or change. So we'll submit our job here. Again, it goes to our job list. I'll click on the job list here and is running. So basically what the Meta-CATS will provide you is it will provide you with a sheet which should have the positions which are significantly different between the two groups, human and swine, and their p-values and what sequences are present in each group. So you'll see it's fairly quick, but our data set is also small. That's the reason. Anyway, so we select the job, and as previously, we'll view it. And as you can see, there are lots of output files. You can go look inside, but what is most useful is this view. This gives you the consolidated report, or for the most time, this is what you want. So you click on the view and you see this is what we got. We got the chi-square value, we got the p-value, and you see it's only significant ones. We got the degrees of freedom fewer than 5, and we have the 2 host groups and the sequences for each one. So see the positions, like for everything, the humans have 1 residue in that position, and for the swine, it's mostly 18 and 3. Remember, the 3 probably are those which lies in the human branch from our phylogenetic tree. See, for the most of it, the 3 swine residues resembles what is in the human group. So another evidence that these 3 strains are more like humans than the swine. And the next step you can take is you can do a multiple sequence alignment of everything. I'm not doing it currently here, but just for the sake of another evidence and just match or verify what the Meta-CATS is coming up with, this change in residue, it's actually true. If you can do that, then there is another evidence.
Let me ask.
Yes, Anna.
So I might have just missed it, but you pointed out the swine proteins that are like the human, but there's some positions that are not discriminate between each, like 394. Or no, that wasn't a good example.
I think you're talking about something like 505.
Yeah.
Anna, this is just a statistical analysis. It doesn't take into account the biology or something like this. So from here, what I will do is take the positions which are significant and then go to the multiple sequence alignment or do a literature search or do other subsequent analysis for each position and see whether it falls under an important domain or motif, or like that can lead to like change in the host or something like that. And also here is just saying 3. But how do we know it's those strains which are similar to the human? So to make it more informational or to get the clearer picture, the next step would be to do a multiple sequence alignment and just verify that this 3, which has a G, are actually those swine isolates or swine strains which are similar to the humans or something like that. Then pick out those which actually differ. Maybe these 3 are separate 3, something else.
So you're using this as a guide for the next step, and not every output of the Meta-CATS is going to be the reason the groups are separated.
This is a supporting segment. If you need a statistical evidence to your results, this can be one of it. But you need other analysis results to support this. This can be a support, but this is not an explanatory thing. So here comes the end of the Meta-CATS. Let me do the structural comparison and then I'll come back for any questions later when I finish. But feel free to put your questions in the chat box if you want, and we can revisit later. So the next one is the 3D structure viewer. The 3D structure viewer we use in BV-BRC is MoldStar. So let's see a demo. Here also I'll take a case study and I'll compare the receptor binding domain within the spike protein for the Wuhan strain, SARS-CoV-2 Wuhan strain, and the XBB 1.5. Little bit background so that you know. So the main difference between these 2 strains in the receptor binding domain is there is a point mutation at the position 486, which is like the XBB 1.5 is defined by that mutation. So in Wuhan, we have a phenylalanine F, and whereas in XBP1, we have proline in that position, and structurally what we can find out from that. So let's do. So here I'll do the search from the featured virus page. So we'll have the SARS-CoV-2, we'll click on it, and then we'll go to the protein structures. And BV-BRC currently supports predicted structures using AlphaFold. We are predicting it ourselves and also pulling from the AlphaFold database. So like you will see each cycle or somewhere in the near future, you'll see more predicted structure in BV-BRC. So here our XBP1 spike protein will be a predicted structure. I wanted to include that so that you know you can do the same things with the predicted structures too. So I'll do some filters because it's lots of structures here. So we'll take the predicted one. We'll take the X-ray diffraction one. And now we want the spike. So we'll introduce the gene field here. And let's find the S here. You can click on each column to sort it here. So I got my predicted one. So definitely I'm selecting this. It's the receptor binding domain of the XBP 1.5. And the other one I'm looking, I previously looked, I know what I'm interested in. So that's the 6MOG. So you can select up to 10 structures here and use this view structure button to click, and it will take you to the MolStar where you can visualize the structure. So before selecting both the structures, I want to show you some functionalities, and that will be better with one structure. So let's start with the 6M0J structure, and then we'll add the predicted structure later for comparison. So I selected my desired structure, and now we'll open the structure in the MolStar. So you can see this is our structure. So something about the MolStar viewer page here. So this is our information panel, you can say. So you can see more details about the structure it brought in, and data is available on a particular structure, like more metadata. You'll see a feature panel like this, where you can visualize this particular areas or domains within the structure. I'll give you an example. Say receptor binding motif. See, when I click on it, it highlights the blue area, and this is the receptor binding motif for the spike protein. And it has the epitopes too. So if the data is available, then you will see this feature here, and you can select, see the yellow one highlighting each epitope area. If you want to visualize, you can select more than one and visualize those. And this panel is the sequence panel. So here it will show you the number of structures that are open here. For now it's 1, so we have it. And then within the structure, you can select the chains, polymers, or anything. It will show the other one. So here we have the ACE2, the green one, which is the human receptor for the SARS-CoV-2. So we have that. We have the spike protein, and we have the other chemicals in it. Once you select a chain, you can select if there are multiple chains and select from here. This is the console. If you do a superimposition or anything else, it will give you the information, the values, or whatever functions you are doing here. It gets activated here. This one has the tools. So this is about the structure. If you take a measurement here, will come here later. This is the components. So polymer, carbohydrates. If you want to hide the carbohydrates, you can hide it with this eye. You can hide the water. You can hide the ions to make it cleaner, or based on your need. The polymer is the whole thing. Now comes to this panel. This is our tool panel, you can say. So the most important is this toggle one, this arrow. So this helps us select particular things within the structure which we want. So once you click on it, this appears, and then the residue. If you want to select one residue or multiple residues which are not like one by one, then you select this. You can select a particular chain, a model, any structure, any residue instances. Like what I mostly use here is the residue chain and sometimes the model. but the most useful is the residue and the chain. This currently, the MolStar has this. You can select one residue and then do this. It will select all other except that residue. It's basically Venn diagrams showing you can select union or intersection between those and something like that. This you can color the selected residue, and this is what you save that component. If you want to save that selection, you can do it. So let's see. Say I want to select few residues within the receptor binding domain that I know is important. So we'll go to the residue because we want to save residues, and say we'll select our residue 486. See, I told you it's a phenylalanine in Yuhan. You can see information here about what amino acid it is, what its position, to make sure you are selecting the correct one. You can see it's a phenylalanine at the position 486 authentically, but in this structure, because they crystallized only the receptor binding domain, here it's 186, but in the spike it's 486, and you click on it to select it. You can see here it's selected. If you want to select multiple, hold your shift button, click again, it selects that whole thing. If you don't want, then it's one, and then you click on another, no holding space, click on another, it will select those. And say these are the three residues which I'm interested in. How do you save it? So you'll go to your cube, click on the cube. It's our current selection, and I want to save it. So now it's a cartoon representation, right? We cannot see the side chains. I want to see the side chains because this is the amino acids I am interested in. So in the representation, we'll click and we'll do ball and stick. And then the option, you can label it to a different thing. I'll say important, and we'll create the component. And once I create the component, you can see the side chains appear. And we have a named important component within your component panel. You can do the same thing. You can hide, show, and do some maneuvers. If you want to change the color, it's already selected. You go to the paintbrush, select a color, and then apply. And you see those are colored. This is for better visualization and anything you want to do. Another feature of MolStar is you have to keep the selection to residue or chain, whatever you want to. But now we want to see deep inside what each residue is doing. So we'll keep the residue and close the selection panel. And if you want to see the interactions, then you click on the residue you're interested in. So for us, you can click from the structure. You can click from the sequence panel. You can click here. So I'm interested in the 486. So I click on it and you can see its interactions. So currently it's not interacting with S2. This is our 486, and the residue after that, the 487, it interacts with the ACE2, and this has some inter-chain stabilizing interactions to maintain the structure. So that's it. That's you want to know. Now we'll go into the case study. We'll open both structures. So we'll close this. And now we have this structure selected, and we'll open our predicted structures. And then 2 items selected. You can open up to 10, but I don't think anyone will do that because then you won't understand anything of the structures. So click on the structure viewer, and you can see now 2 structures. Like one is previously our ACE2 and our Wuhan. And now you can see another one appeared, purple one. This is our XBB 1.5 spike. And you can see here, now we have 2 structures because 2 structures are open. Now, how to compare? You need to superimpose the structure, align the structure together, and then only you can see the difference in the structure, right? Once you open multiple structures, there is another option which appears is the superimposition. So to superimpose 2 structure, you need to select 2 similar chains. Obviously, you don't want to superimpose a spike protein with the ACE2, right? So we need to select the 2 spike protein chains. So from residue, we'll now go to the chain, and then we select the chain. You see it got selected. So this is for the Wuhan, the 6M0J. And now we'll move to the other structure and select the other chain. You could have done it from this console too. You could have clicked on 2 and it would have selected. You can do it this way too. So now the 2 chains are selected. You go to the superimposition option. You go to the chain option. You can see the 2 chains which you selected, and we click on superimpose. Once you do that, look in the console area. You'll see that superimposition happened with the RMSD of 0.74. RMSD less than 1 is good. That means the 2 structures are quite similar, like almost no difference. Now you can see 2 chains superimposed. So one is violet, another is reddish maroon or something like that. And if you look closely, let me change this to residue because it's selecting. Yes, now it's better. You can see there are minor differences in the loop areas here and there, but mostly they are similar structure. They have the similar fold. I'll switch off the carbohydrates, water, ions. Okay. See, there is minor difference in the receptor binding motif. You remember the receptor binding motif? We can select from here. You see this highlighted area. If you look, there are minor differences, but can this minor differences cause any change or not? That's what we are going to look into. So the next thing is what we are looking for is the position 486. So we'll select position 486 for each state. So let's start with the Wuhan. The ACE2 is open. We'll go to the spike protein and find the position 486. Okay, position 486. We select it and then we save it with the side chain and label it Wuhan and create a component. It will help us come back to it. Then we go to the other, I think it was 168 here, right? Yes. And then here we'll look for the 168. Look, it's a proline. Remember the mutation was from phenylalanine to proline. This is not giving the authentic number because it's a predicted structure and we only use the receptor binding domain. So the numbering, the amino acid numbering is only for the receptor binding domain area. So we'll select on the proline and select it with the side chain option. We'll write XBB and create component. And now we have 2. Let's focus on it and you'll see it's quite different. It's creating a change in the backbone structure. So let's focus on it. Will this mutation change anything in terms of the interaction profile? So to know that, we'll close the selection panel. We'll click on each residue to look for its interaction profile. So let's start with Wuhan because we have more information on it. So once we do that, you'll see as you looked previously. So the Wuhan, this is the phenylalanine, this is the position 487, which interact with the ACE2, that the human receptor at 2, like it makes 2 interaction, 2 bonds with the human receptor and 2 bonds with itself, with the backbone structure. And what will happen if we click on the proline? You will see that due to this mutation, there is a shift in the N from here to here. So you can see due to this position, the side chain of the N changed position, but it introduced an extra stabilizing bond within itself. This makes this receptor binding motif more stable. It introduces one extra bond compared to the Wuhan. But here we cannot predict whether by just seeing I can say, okay, it will definitely lose this interaction because it's too far away. But I'm not sure about this. But just from here, we cannot say whether any change will happen to the interaction. You have to do an interaction analysis, which is not currently supported in ModeStar. It's just a viewer. But you will have an idea that you can draw a hypothesis that due to this change, there is a shift in the ACE2 interacting residue within the spike protein. So probably it will affect its binding with the ACE2. And apart from that, it introduces an extra stabilizing interaction within itself. So the next step with this will be you do an interaction analysis. You predict the interactions between the Wuhan and ACE2 and the XV1 and ACE2 and see whether there is change in the number of interactions. If you are interested, curious, there is a difference between due to this mutation, This has only one interaction with the ACE2. It only interacts with this one and not this one. But you have to do it separately. So one thing you can do is you can go to other software which does this, or you can come back to Moldstar after you perform a dock with the XBB spike protein and X2 and then visualize it using this method. So this is most of the functionality that you have in BV-BRC Protein Structure Viewer. So that's it from me regarding this. If you have any questions, please feel free to ask.
Thanks, Roshni. One question, how accurate is the structure prediction?
It is quite accurate. Just to test, I have predicted the sequence of a already crystallized structure and tried to compare it, see how different or how similar it is. The RMSD was less than 0.5, so it's quite similar. But something to keep in mind that it's still predicted. You can draw a hypothesis or you can say that we are thinking about this, but this cannot be the ultimate thing. But currently, we have so many sequences and so less structures. So this is the one step towards assisting you in your work, because that becomes a bottleneck if you want to do a structure related or anything about the structures, because most of the structures are not crystallized. There are difficulty crystallizing. The predicted epitopes, where did you see the predicted epitopes? So the epitopes I was showing, those were pulled from IEDB. And if we can map a IEDB position on the structure, they have the same sequence, then we show them. But for the predicted structure, we don't do any prediction about whether a epitope will fall in that predictive structure or this will be epitope within the predictive structure. So nothing is shown within the predictive structure because it's predicted and we don't have any evidence of whether it will be same or not. So nothing is shown on the predictive structure.
Predicted epitopes, are they based on human T-B cells?
Yeah, any epitope information you see will be pulled from the IEDB. That's why we don't do any mapping on the predicted structure. We just leave it in case user wants, they can do it on their own. But we are not doing that.
We're always available at help@bvbrc.org. You can also fill out the contact us form on the website if you ever have any trouble with a job or just have a question about features. But thank you all for coming. But have a great rest of your day. And thank you again, Roshni, for a wonderful presentation.
Thank you.